The morning sun pierced through the bamboo slats of Antonio Sablan's kitchen window, illuminating the glass of water that slipped from his fingers and shattered on the coral stone floor. His wife Maria rushed forward, but Antonio's left hand seized the table edge, knuckles whitening as his leg buckled beneath him. Dr. Manuel Cruz pressed his stethoscope against Antonio's chest in the cramped clinic, dismissing the tremors as shell shock from Japanese occupation. Yet 3 villages away, fisherman Jose Taitano collapsed at his nets, words dissolving into unintelligible slurs. Within weeks, 17 more Chamorros stumbled into island clinics, their symptoms identical. The American Naval Hospital's logbooks filled with the same clinical observations: progressive muscle weakness, dysarthria, fasciculations rippling beneath sun-weathered skin. Nurses watched Antonio's fingers twitch uncontrollably as he struggled to sign his name, the pen dropping twice before his signature devolved into an illegible scrawl. Antonio's case multiplied across Umatac and Agate. 12 victims by March, 37 by June. Dr. Eleanor Vance pressed her stethoscope against another patient's chest. The heartbeat strong, the lungs clear, yet the man's legs refused to obey. She watched his fingers curl involuntarily, the tendons beneath his skin tightening like piano wires. The hospital's 32 beds overflowed into hallways where makeshift cots held trembling bodies. Vance's pencil scratched across identical charts: muscle rigidity, cognitive decline, no fever, no inflammation. She drew blood from a fisherman whose speech had dissolved into whispers, then from a grandmother whose hands shook too violently to hold her rosary beads. Laboratory results piled on her desk: negative for polio, negative for encephalitis, negative for syphilis. Under her microscope, cerebrospinal fluid appeared pristine, no white blood cells swarming to fight infection. Outside her window, ambulances continued arriving, their red lights painting the hospital walls, while inside, the paralyzed stared at nothing, trapped behind eyes that no longer blinked. Dr. Eleanor Vance's urgent cables crossed the Pacific, landing on desks at the National Institutes of Health. Within weeks, a C-54 Skymaster touched down at Anderson Air Force Base, its cargo hold packed with centrifuges, microscopes, and America's brightest epidemiologists. They established their laboratory in a converted military barracks where ceiling fans churned the humid air above rows of gleaming equipment. Blood vials multiplied across refrigerated racks, crimson samples drawn from trembling arms, labeled with Chamorro names the scientists struggled to pronounce. Dr. Marcus Chen hunched over his microscope for 16-hour stretches, scanning slides for viral inclusions that never materialized. Spinal taps yielded clear fluid, devoid of the cloudy infection markers that would explain the paralysis creeping through the villages. Petri dishes lined the incubators like glass coins, their agar surfaces pristine after weeks of cultivation. The geneticists sequenced chromosome after chromosome, finding only normal Pacific Islander markers. By November, Chen's notebook contained 347 negative results. The disease-killing Guam's people left no fingerprints, no bacteria blooming in culture, no virus hijacking cells, no inherited mutation warping DNA. The Americans departed with their microscopes and empty test tubes, leaving behind a name for the terror, Lytico-Bodig. Inside the cramped hospital ward, 63-year-old Manuel Borja gripped the metal bed rails, his knuckles whitening as another spasm seized his right leg. The muscle contracted violently beneath paper-thin hospital pajamas, twisting his foot inward at an unnatural angle. His daughter pressed a wooden spoon against his lips, watching rice grains tumble onto the starched sheets. Manuel's jaw clenched and released in erratic rhythm, each attempted swallow triggering a cascade of facial tics. The man who once hauled 100-pound nets from dawn waters now struggled to command his own tongue. His eyes, still sharp with intelligence, tracked every fallen grain. Down the corridor, similar scenes multiplied. A former schoolteacher's fingers froze mid-sentence while writing a letter. A carpenter's hands locked around phantom tools. The paralysis advanced methodically. First the tremors, then the stiffness, finally the silence. Families crowded the hallways, watching their elders disappear inside bodies that had become prisons, muscles betraying a lifetime of practiced movements. The American scientists packed their microscopes into aluminum cases, their months of fruitless investigation leaving only exhaustion and unanswered questions. Outside the cramped laboratory, Guam's volcanic soil stretched dark and rich beneath towering coconut palms, its mineral composition unlike anything found on the mainland. Dr. Vance knelt beside a taro patch, her gloved fingers scooping the black earth into sterile glass vials. Machetes sliced through vines as thick as ship rope, while botanists catalogued every leaf, bark sample and root system that touched Chamorro dinner tables. An elderly woman named Maria watched from her doorway, her weathered hands clutching a walking stick carved from ironwood. She beckoned the youngest researcher closer, speaking in halting English about the orange seeds her grandmother once soaked for days in running water. The cycad trees stood ancient and unbothered at the jungle's edge, their crown of fronds sheltering clusters of seeds the size of walnuts. Maria demonstrated the old preparation method, crushing, soaking, draining, her fingers mimicking the careful movements that separated sustenance from poison. Without proper washing, she warned, these seeds could kill within hours, a fact every Chamorro child learned before they could properly hold a fishing spear. Maria's weathered fingers traced the cycad tree's rough bark, the same poisonous giant her grandmother had warned against decades earlier. The orange seeds hung heavy from ancient branches, each one the size of a child's fist, their waxy skin gleaming under Guam's relentless sun. Scientists collected samples, unaware they held a toxin that had killed for centuries. Chamorro women traditionally soaked these seeds for days, pounding them into flour after countless rinses drained the poison away. One mistake meant instant agony, vomiting, convulsions, paralysis striking within hours of consumption. Laboratory analysis revealed BMAA, a neurotoxin so concentrated that a single unprocessed seed could kill a grown man before sunset, its molecules attacking nerve cells like microscopic assassins. Yet the dried flour samples from village kitchens tested nearly clean, the toxin levels too low to explain the devastation ravaging the island. Dr. Vance stared at the data, her pen tapping against contradictory results. Acute poisoning struck immediately, but Lytico-Bodig victims had eaten cycad flour for decades without incident. The answer lay elsewhere, hidden in plain sight. The cycad mystery had consumed months of fruitless laboratory work, but Dr. Paul Cox chose a different path. He climbed into the jungle canopy at dusk. His binoculars tracked dark shapes against the orange sky. Massive fruit bats, their leathery wings spanning 3 feet, descended onto the cycad trees. They gripped the toxic seeds with their clawed feet. Cox watched the bats tear into the bright orange flesh, their sharp teeth crunching through the poisonous pulp that would kill most mammals within hours. Below him, a Chamorro hunter named Vicente whispered the local name, Fanihi, and explained how his grandmother taught him to net these creatures. The bats hung upside down from the cycad branches, gorging themselves on seeds containing enough BMAA to paralyze a human. Vicente demonstrated the traditional preparation, the captured bat, still alive, thrown whole onto glowing coals, the fur singed black, the skin crackled, and the meat beneath roasted in its own fat that had accumulated years of neurotoxin. Every wedding, every funeral, every celebration featured platters of these roasted bats, their small bones sucked clean by honored guests. Dr. Cox's discovery sent tissue samples from the captured bats to mainland laboratories, where technicians in white coats sliced paper-thin sections of wing membrane and liver. The fruit bat sprawled on the stainless steel table, its leathery wings pinned open, revealed nothing unusual to the naked eye. No tumors, no lesions, just the ordinary anatomy of a flying fox. Under the microscope, adipose cells bulged with yellowish droplets, each one a microscopic time bomb of concentrated BMAA. The neurotoxin had infiltrated every gram of fat, binding to proteins and accumulating year after year as the bats gorged on cycad seeds. Chemical analysis revealed concentrations 100 times higher than in the seeds themselves. Bioaccumulation at its most insidious. The bats thrived, their nervous systems mysteriously resistant to the poison that liquefied human neurons. They roosted in the jungle canopy, groomed their fur, nursed their young, all while carrying enough neurotoxin in their bodies to paralyze a dozen men. Nature had crafted the perfect vessel, a living, breathing reservoir that transformed a plant's defense mechanism into a decades-delayed death sentence. The yellowish fat globules discovered in the laboratory had concealed their secret for decades. Inside Antonio's brain, the BMAA molecules began their silent infiltration, slipping past the blood-brain barrier like thieves in the night. The neurotoxin mimicked serine, an essential amino acid, fooling the neurons into accepting the imposter into their very structure. Each contaminated protein twisted and misfolded, triggering a cascade of cellular destruction. The motor neurons in the spinal cord withered first, their axons retracting like dying vines. The substantia nigra darkened and shrank, dopamine production grinding to a halt. In the temporal lobes, tangles of corrupted proteins strangled healthy tissue. The poison worked with methodical cruelty, dismantling neural pathways built over a lifetime. Synapses that once fired in milliseconds now sparked erratically, then fell silent forever. The brain's electrical storms dimmed to flickering embers as BMAA molecules continued their relentless substitution, transforming living neural networks into graveyards of dead cells. Dr. Paul Cox's revelation transformed yellow cycad seeds and roasted bat meat into weapons of molecular destruction. The pathway emerged with brutal clarity. BMAA molecules, forged in the cycad's tissues, concentrated 100-fold in bat fat, then lodged permanently in human neurons after a single feast. Each wedding celebration, each honored guest served crispy fanihai, unknowingly consumed a time bomb. The neurotoxin's mechanism defied conventional poisons. BMAA masqueraded as serine, an essential amino acid infiltrating protein synthesis within brain cells. For 20, 30, even 40 years, these counterfeit molecules accumulated silently until critical mass triggered catastrophic failure. Motor neurons withered, speech centers collapsed, memory dissolved like salt in rain, leaving behind the hollow shells of Lytico-Bodig. Antonio's trembling hands, Maria's frozen stare, the fisherman's spilled rice, all traced back to communal fires where grandparents had savoured sweet bat flesh decades earlier. The Chamorro's cherished tradition had become their curse. As Fanihi populations dwindled and consumption ceased, New cases vanished, but hundreds remained trapped in bodies that would never heal, monuments to a poison that travelled through time.